in dermatology over the last decade, it's become very apparent the importance of interleukin 23 to plaque psoriasis pathogenesis, also to psoriatic arthritis pathogenesis, although certainly more to the plaque psoriasis. And what we've realized is that as there's been an evolution in generations of IL-23 targeting biologics, that's a progression from targeting really the P40 subunit, ustekinumab, to the P19 specific subunits of IL-23, which are Risankizumab, tildrakizumab, and Guselkumab. So this evolution of how we target interleukin 23 has really become clinically apparent as these IL-23 biologics, IL-23 targeting biologics have come to really dominate the clinical realm of psoriasis treatment. So what my laboratory really wanted to do was to better understand the differences in these IL-23 biologic inhibitors. So we know IL-23 is so important to psoriasis pathogenesis, and we've gotten accustomed now to numerous products on the market. As I said, the ustekinumab, which targets the P40 subunit. So we have to remember IL-23 is made up of two subunits, the P40, which was the first generation target by ustekinumab, and then the P19 component, which is really the second generation where you have the Risankizumab, tildrakizumab, and Guselkumab targeting it. And we've been using these medicines for the last several years, really seeing tremendous results from all of these medicines in our patients. They are absolutely superior to no treatment at all. That is a given. But what I noticed in taking care of my own patients was, what are the differences between these medicines? How do you know what to choose? How do you choose among these different options of IL-23 targeting biologics? And I don't think there's a lot of information out there to really know how. There were certainly some network meta-analyses done that kind of looked at which of these medicines performed better in terms of clearing the skin. What we know is the PASI response, right? PASI 90, PASI 100. And so when you look at these network meta-analyses and you get an idea, okay, well, these particular medicines seem to have the greatest PASI 90 or PASI 100 response. And at least in the network meta-analysis we used in our study, that drug was Risankizumab. What we wanted to do was to say, can we take the molecular properties of the drugs and their binding target on P19 and make some type of correlation or understanding of the link between the chemistry and the structural biology of IL-23 to the inhibitor and its clinical function or clinical outcomes for the patients. And I think that in dermatology, this type of research connecting structural properties of medications and their target to actual clinical outcomes hasn't really been done before, at least not in the way that we did it, and certainly not for P19 targeted biologics. And I really was hoping that this project would be able to provide more clarity to providers of what these medicines are and what are the differences among them. Because I think that many dermatologists may still lump all of the IL-23 biologics together. When you talk to your patient, hey, this is an IL-23 inhibitor or an IL-23 targeting biologic. And that's the limit of our understanding or discussion. And what we wanted to show in this paper that was published in JID Innovations is that there is a very rich molecular basis for how these medications function and the differences between them. And that was the overall premise or idea behind what we wanted to do, was really say, what is it that's unique about where these drugs bind on P19? And what is it that maybe the features of that binding site, which we call the epitope, what are the features of that epitope on the P19 subunit of IL-23 that may actually give some level of clinical response using the metrics that have been previously done in network meta-analyses? We often hear clinicians talk about binding affinity. this drug has better binding affinity than this drug. And certainly when you think about IL-23 targeted biologics, you do think, okay, well, this drug may have higher binding affinity than this one. Okay, but what does that mean? And I think it's important for dermatologists to understand that the binding affinity is a composite of actually two factors. So when you think of binding affinity, it may or may not be just good enough to think of binding affinity by itself. Binding affinity has what's known as the K-on rate, the rate in which a complex assembles, and it has a component of the K-off rate, that the rate in which that assembled complex, in this case, the IL-23 targeted biologic bound to IL-23, the rate in which that complex dissociates. And we really wanted to understand, well, which of these features, these structural and molecular features of the P19 epitope, where these biologics are binding, which of those features actually correlated with clinical response? So what we found was very interesting. We found that the epitope surface area actually does matter. It was very important. And so the larger the surface area of the epitope seemed to correlate with two things: a higher overall composite binding affinity, but importantly, a very better, stronger, and in this case, lower K-off rate, right? So a stronger complex that lasted longer, that came apart slower. And so that larger surface area correlated with a lower K-off rate, meaning more stable long-term IL-23 targeted biologic IL-23 complexes. And so what was really fascinating is that when we looked at the different IL-23 biologics, that there was certainly a difference in surface area among the epitopes. Risankizumab had the largest, followed by Guselkumab rather closely. And then much lower down, you had Ustekinumab and then Tildrakizumab. In the clinic, we know that Risankizumab has been a very strong performer, if not the best performer, of the P19 specific biologics in treating plaque psoriasis. And what we feel that our paper, our analysis has done is to shed molecular insights into what really makes Risankizumab different as a P19 targeted biologic. I think there's a few key factors. The first factor is it's binding a very key loop region that no other biologic is targeting. Second, it has the largest epitope surface area, and that matters because it has the lowest K-off rate. That means Risankizumab is forming the strongest complex, drug or biologic complex with P19, with the IL-23. And that, I think, has clinical impact because if you're soaking up IL-23 with Risankizumab and it's not coming apart, that complex is not coming apart easily, that IL-23 can't signal, that IL-23 can't perpetuate the signal that's driving psoriasis. And so these are features we really want in the therapeutic, and Risankizumab does it very well. And I think that when you look at our paper and our data, you have Risankizumab and Guselkumab that really separate away from all the others. And the Risankizumab itself, as I said, has very neat properties compared to Guselkumab at that. And I think that this is a very, very enlightening study. And I'm super excited because when I'm in clinic and I'm taking care of psoriasis patients, I'm thinking about some of these discoveries. Oh, wow, this medicine does this. I think this might be a really good one for you. I think about it this way. I don't know if all dermatologists are thinking it blanket that way. But I do think that this type of molecular understanding and really understanding why is it that a medicine may be highly efficacious, it's really important. And this, certainly for Risankizumab, explains a lot of why this medication has become one of the most popular, if not the most popular prescribed biologic for psoriasis at the current moment.